Hi everyone, welcome back to Neurobiology at Providence College. I'm Joe DeGeorgis. Hi everyone, it's Joe. It's hard to believe that this is our last lecture for PC Neurobiology for 2020, but here we are in our online format. Today's topic is about synaptic connections, and it's particularly interesting, I think, because memory is thought to be stored through synaptic connections. And of course, you might be able to remember something from as far back to when you were 3 or 4 years old. And that memory doesn't have to be traumatic or anything like that. It can be pretty mundane, but you can remember it. And at the same time, you might forget where you parked your car coming out of school or something like that. So there's a long-term memory process, and then there's short-term memory, and we don't know very much about either of those. Although generally people think that memory occurs through synaptic connections, which is why this paper is so interesting. So just to give you an example, you can have two neurons that are next to one another, and they might not have a synapse, but somehow through experiencing something, those two neurons might start to interact in a sort of mild way with a transient or a synapse that starts to form. And the idea is that the synapse might get stronger over time until those two neurons are connected permanently. And people might say that that connection is somehow associated with memory. So, to give you a real example, I took piano lessons when I was a kid, and you'd practice, and then you'd practice and practice and practice, and you'd get better and better and better, and maybe that's because the neurons are forming specific synapses, and then every time you practice, they get stronger and stronger and stronger, and then once they're hardwired, those synapses don't come apart. So now, even though I learned that a really long time ago, and I can no longer read sheet music. I mean, I know FACE and all that sort of stuff. I can figure it out, but I haven't read music in a long time. But I can still play a half a dozen songs or so because, well, they're hardwired into my brain. I mean, I'm not great at it, but I can do it, and I'm sure if I practiced, I could get back to where I was. And that's the general concept of how people think memory works, through synaptic connections and the strengthening of those connections. This paper, though, argues that some of the synapses are transient, that meaning they might form and then they might disappear. And that is a pretty novel concept. Most people wouldn't have thought that that was the case. It turns out that the paper was written by a friend of mine, Karel Savoda, and he's at Genelia Farms, which is a part of Howard Hughes Medical Institute, HHMI. And at any rate, there's just a few figures that I want to go over. This will be a little shorter than yesterday's lecture on color blindness. But at any rate, if we take a look at, let's see what figure this is. Must be figure 1. Yeah, actually figure 1 is the only figure that I want to go over. So if you look at figure A, 1A, they say PND, which is postnatal day 14, and the bottom one says postnatal day 28. And I'm sure you know that that means the number of days after birth, postnatal day, whatever, 14 or 28. And what they're looking at here is a transgenic mouse that they made. So they took a gene for YFP, which is like green fluorescent protein, except it's yellow fluorescent protein. So it's a fluorescent molecule, a gene that makes a fluorescent molecule. And they inserted it into the genome of the mouse. And they are expressing this particular yellow gene, and what they're showing is in figure 1A. Let me see if I can erase what I have here. They show these two figures, that is postnatal day 14, postnatal day 28, and this is a cortical section they say, it's in the figure legend. So it's the brain of these very young mice, transgenic mice, and P and D 14. So 14 days after birth, postnatal day 28. Okay? And what they're looking at is YFP expression, yellow fluorescent protein expression in the brain. And in the first figure, the brain slice looks a little bit like this. And then you can see that there's this dark band that's like this, something like that, okay? And what the black band is the fluorescence. So this is a little bit complicated. They are using a black and white image instead of a color image to show the fluorescence, because you don't have to publish it in color. So it's bright light that's being emitted, but it's a black band. What they did is they photographed it with a camera, they turned the image into a black and white image, and then they reversed it. They inverted it. So stuff that is black is bright, and stuff that is white is dark. Okay? Or sorry. Stuff that is bright is black, and stuff that is not giving off light, which would be black, is now white. So what they're showing here is the expression of YFP in this 14-day-old baby mouse. Okay? And then in the next figure, you can see that there's a lot more black. There's different bands here. There's a wide band like this, and some more down here. And again, the dark is the bright stuff, and the white is the dark stuff, or stuff that is not putting out any light. So at 28 days, there's a lot more of YFP expression. Okay, and what they did for this is take the brain out of the organism and then slice it into thin little sections, and then look at the section, put the section on a slide, and visualize this section to see the fluorescence. Okay, so they're just trying to show you that the YFP is being made, and as the organism matures, more neurons start making YFP. Of course, to do this experiment, they had to use two different mice, because one is 14 days old, and they had to sacrifice that mouse for this brain slice, and then they had to use a second mouse that they kept for 28 days to obtain the second image, the postnatal day 28 image. Okay, so for panel B, figure 1B, what they did was something I thought was really awesome, very cool, is they took a single mouse, and you know what a great artist I am, so I'll draw the mouse for you. And oops, what happened? I'm in eraser mode. Okay. And what they did is they put the mouse under anesthesia, and they cut this part off. That is, they remove the scalp, the bone. So I'm going to take this and I'm going to erase this part. So they cut that off, and then without hurting him, her, too much, they put a glass cover slip on top of the mouse's brain. So the brain is here, and now it has a microscope slide that's been superglued to the top of the mouse. So now they could take a microscope objective, like a 100x objective, and they could peer inside the mouse's brain and look at fluorescence. And in figure B, 1B, they show the mouse at postnatal day 16. And you can see a dark area, which is the brain, and then you can see some nerve cells which are fluorescing. So the lines that you see are individual neurons that are expressing YFP, and the protein is made, and then it floats around in the cytoplasm and it fills up the volume of the nerve cell. So this image is not inverted like figure 1a. The stuff that's bright are indeed the neurons. And it's important that not all of the neurons are expressing YFP, because otherwise all you would see is white. The whole brain would be white. Of course, I've drawn it as dark lines, but take a look at the figure and you'll see what I mean. The bright lines are some of the neurons of the brain expressing yellow fluorescent protein, and they removed the color, so it's just a black and white image. So the white lines are indeed the expression of YFP. And then if you see at postnatal day 25, which is the next figure over, so they take this mouse, they strap him down somehow so that they can look at the brain, and then they take the mouse, under anesthesia, but then they take the mouse off of the microscope, and he recovers, she recovers, and runs around until postnatal day 25, and they put the mouse back under anesthesia, and they find the exact same part of the brain, and this time you can see tons and tons of YFP expressing neurons. So just like in the first figure, the older the organism gets, the more YFP expression you see, and they're just demonstrating that they are indeed expressing the protein and that you can see it, and you can take a look at the mouse and then let them run around for a while and then look at them later in time and see this change in expression. That's just the first part of the experiment. The cool part is figure C, so let's get to that. Okay, so figure C just shows one single nerve cell at very high magnification. So this is figure C, and they're looking first at postnatal day 16, they say, and you see this structure with, the nerve like this, and then, if you draw a synaptic connection, which we've done in the past, the presynaptic terminal, that this is one neuron, the upstream neuron, is filled with synaptic vesicles. We've said this before. So this is part of one neuron, and then the postsynaptic side of the synapse has a postsynaptic density, which contains the receptors for the neurotransmitter that stick out from the cell membrane. They're transmembrane proteins, so they are partially inside the cell, partially outside of the cell, and partially within the membrane itself. And we know that these fuse with the cell membrane, the recent Heuser paper, and they release neurotransmitter, and then this neuron, downstream neuron, fires if it receives enough of a signal to reach threshold. And anyway, if you just look at one, so if this cell here is lit up and this one doesn't have YFP in it, so it's totally dark, all you see is one of the two cells, the one that's expressing YFP. And that's what they're trying to show you here, is that you see these little structures like this, and these structures are these structures. So one neuron synapses many times with other nerve cells through a synaptic connection like this. But what I'm trying to get at is that you're only looking here at one cell. So this here is called a bouton, and you're looking at one half of the synapse, either this part of the synapse, the postsynaptic terminal or the presynaptic terminal. So that's what you're looking at. So they're looking at these synaptic boutons, as they're called, okay? And then they have postnatal day 17 and postnatal day 18 and so on, all the way to, let me see, I have to look at the paper, 20, all the way to day 25, it looks like. Yes, all the way to day 25. And what they're trying to show you is some of the boutons, which are synonymous with the synapse, one half of the synapse, appear and remain steady over time, over the full 16 to 25 day interval that they're looking at. And those examples are in yellow. So if you look at the yellow arrows in postnatal day 16 at the top, you can see that all the way through day 25, you still see that bouton, which means those synapses are extremely stable. So they show, you know, they've got one up here with it, they got an arrow like this and an arrow like this, and you can see that all the way through day 25. If you look at the blue, though, in postnatal day 16, you see a structure there, and it starts to disappear in postnatal day 17, and then it's completely gone by postnatal day 18. And if you look at the red arrow, which starts in postnatal day 19, it's pointing at a bouton, and that bouton gets bigger, and then it stays for the remainder of the time period. And then finally, if you look at the blue arrow in postnatal day 22, you see something start to form, a bouton starting to form, and then it's there for day 23, day 24, and then it's gone in day 25. Okay, so now if we look at the title of the paper, it's Transient and Persistent Dendritic Spines in the Neocortex in Vivo. And dendritic spines, let's talk about that. We started our class actually talking about dendrites, and we said that, you know, the neuron contains a cell body with a nucleus, and then it has the axon like this, and then the terminal end of the cell, which synapses with other neurons or connects to muscle through the neuromuscular junction, and then the branches of the trees up here are the dendrites. And dendrites receive information, so they contain these postsynaptic densities. So you have the boutons are on the dendrites here like this. So the boutons that show up on the dendrites are postsynaptic terminals, and those that show up on these terminal branches are the presynaptic terminals that contain the synaptic vesicles. So that's what the dendritic spine, these are dendritics, they also not only call them boutons, but they call them dendritic spines because they have these little, you know, they kind of make them look spiny. I mean, they're round, so they don't look sharp. But anyway, you saw the pictures. So these are the dendritic spines, that's what they mean, which is what we're talking about here. This is a dendrite, this nerve cell is a dendrite, and these are dendritic spines, or these boutons, as I mentioned. And neocortex obviously is the brain, or part of the brain, and then in vivo means that, you know, in the organism. So the mouse was alive, and they were just peering inside the brain with their cool little cover slip technique. So, and the point of it is that some of them are persistent, and that's normally what people think of when they think of synaptic connections, that they start really weak, and then as an experience happens more frequently, that maybe there's stronger and stronger synaptic connections, and that those connections then are permanent. But what they're showing in this paper is that not all of them are permanent. Some are permanent and some are transient. That is, they can form, stay there for a while, and then they can disappear. Now, whether that's, you just lost a memory or not, I don't know. That's a little bit more of a complex issue. I also think that there must be elements to memory that don't specifically involve synaptic connections, because you might have an experience where it's not like practicing the piano. You're not doing it over and over and over again. It's just like you walk by someone in the hall one day and you see them, and then 6 months later you come in, you see them again, and you recall the first time that you saw that person. And the memory process still might happen through synapses, but they may be synapses that have already been formed, and there's just some quick biochemical phenomena that happens that allows you to remember that. I mean, we just don't know. The brain is a very, very complex structure. Okay, wow. I can't believe it. I can't believe it's over. I know we have a little bit of studying to do, you guys, and I hope you guys can study. I really want the end of the semester to go well and to hand out a lot of great grades, or all great grades if we can manage it. So we have a little bit more work to do in terms of studying, and I'm going to have a review session, at least one. If you would like more than one review session, I'm happy to do more than one. If you want to Zoom by yourself or with a small group, I'm up for that. If you want to ask me some questions by email, you want to call me on the phone, you want to FaceTime, you want to text message back and forth, whatever means you would like, I'm here for you guys. I'm here to try to finish off this semester in a strong way. I think I said at the beginning of the semester that the brain is not very good. And I'm sticking to my guns on that, or at least mine isn't very good. So maybe I can't speak for anyone other than myself, but your brain makes a lot of mistakes. And we also said that you end up forgetting a lot of what we talked about, a lot of what we learned in the class, and that's not saying anything about us. It says something about our brain and how it works. And we really know very, very little about how that happens, how the brain functions. And I said that my expectations for the class were to give you guys practice in reading scientific literature and to try to develop your skill of doing that, because I think reading these papers or reading scientific literature in general is going to be important for your career if you go into any sort of science or any branch of medicine or biotech or whatever it is. And so I think that is a very important skill to have. And then I said, I hope that at the end of the class, if you already didn't think so, which you probably did, but that you think neurobiology or neuroscience is an interesting topic, and maybe some of you will pursue that. And I certainly hope that that's the case in some whatever capacity that might be. And finally, I would say that I'm happy to write letters of recommendation and help you guys in any way that I can moving forward. And so please lean on me if the opportunity arises. So, okay, that's it, you guys. I will see you in a few days. Have a good weekend. Bye-bye.